So my goal over the last 14 years or so has been to change the practice of medicine. And we're trying to do that by discovering new medicines. When I arrived out of Harvard now into a company, Novartis, shortly after I arrived, they asked me for my strategy. And I said, I've been at Harvard for 25 years. I don't have a strategy. I don't know what strategy is. And they said, well, you have to come up with one in this meeting because the CEO is going to want it. And I had just met, and this is an advice for any of you who are going to go into consulting, I had just met a bunch of consultants. I won't tell you from which firm. And I realized that when they're asked for a strategy, their answer is always the same. It's a 2x2 chart. And it doesn't matter what the axes are. You just have to be in the upper right-hand corner. And that's a successful strategy. So all I had to do was label the axes. And one was a need, an unmet need. This, by the way, is not driven financially. This is driven by a patient who might need a medicine. And the second, is which will be the focus of today, is the disease mechanism understanding. In other words, we have to know the basic science behind a process before we can even begin to make a medicine. And that's even itself not always enough. And it takes a very, very long time between the basic discovery and actually making a medicine. And this is why. Part of it is you can have a strategy, as shown on the left, but a lot of serendipity. These are the three Persian princes of Serendip, which gave the name serendipity. And serendipity is not blind luck. Serendipity is informed luck. So you have your basic discovery, and it winds around through lots of people in many, many years, and somebody at some point realizes maybe we could make a medicine out of it. You say, why does it take so long? I'll give you a sense of that today. But I looked at recently all of the most important medicines and found that the time from discovery, the basic discovery that gave rise to this drug, and the actual release of a drug was minimally 25 years, often 30 or 40 years. So for example, the most standard antihypertensive medicine, an angiotensin-converting enzyme inhibitor, which came out in 1981, traces a long history of understanding of blood pressure control that goes back 100 years. To the discovery of another enzyme that affects angiotensin 1. You say, why did it take so long? I mean, this was obviously important in blood pressure from the start. And I know a lot of people who were involved in this process, they didn't believe it would be safe, or they didn't think it would be good, or they didn't know which part of this pathway to hit. So you have to get used to the fact that the time from the discovery to the drug is very, very long. And this is true for the drugs we use today. But what I want to focus on today are the ones we're going to make for tomorrow. And I'm going to take 3 examples of very important directions that are right now at the basic science stage and just beginning to leak into making drug. I'm going to tell you how we're going to cure cancer, how we're going to fix the genes of people who have genetic diseases, and how we're going to reverse aging. And I'm going to tell you this using work that's so fundamental and organisms that are so weird that you're going to think this is completely irrelevant. Okay? And my first one is, how do we cure cancer? And now this work goes back to the 70s. The fundamental work that led to all of the important cancer drugs today goes back to the fruit fly. The fruit fly in the 1970s, Janninus Volhard and Wieshaus were interested in a very, very beautiful fundamental problem. How does an egg become an animal? Seemed at the time too complex to approach. They said, we're going to knock out genes in the fruit fly, and we're going to look at the fruit fly embryo, the egg, and how it develops. And they couldn't even get funding for it at the beginning. People said, there's lots of ways you're going to mess up this embryo, and it's not going to be interesting. You're going to make a lot of genetic mutations, but who cares? So they did a screen, and they just made a lot of mutations just using a chemical. And then they looked for several years at what happened to the animal. And it was amazing. What they found was that this was extremely informative, that there are genes, which we know about because they're knocked out, that say this is the front of the animal, this is the back. This is where the wing will be. This is how it's going to be divided into segments. And they could put these into classes, and they did. They put them into classes. You can see this is the normal pattern. And then they just looked, and they said, now this is just the cuticle pattern of the fly. They don't know the names of the genes because it was all random, but they know that there are such genes, okay? And then they realized that some of these looked the same, different genes, but they looked the same. And by a variety of genetic tricks, we're able to put them into what is called a pathway. That there is a signaling molecule that binds to a receptor that activates a bunch of genes inside the cell. Okay, beautiful work. Told us how it was working in the fruit fly. Turns out, works the same way in humans. The same genes, exactly the same genes. Slight differences just from evolution, but the same function are in this same pathway. So you've got a ligand, a soluble substance that binds to a receptor and then activates a bunch of things. And it's used many, many times in humans, in many different tissues, but the fundamental pathway is exactly the same. And when it's overactivated, you get colon cancer. And many other types of cancer. But almost every case of colon cancer, which is the second biggest cancer killer in the world, is due to activation of this pathway. Often due to loss of this particular protein. The details aren't important. What is important is this now tells us how to begin to target new drugs. Because if we know it's overactivated, this is what we want to slow down. And now let me show you another example where we have drugs already. This one we're just getting there. We're into the clinic. And this comes from the worm. This is a nematode worm, a couple thousand cells. Lives on bacteria, not a very imaginative organism. And Bob Horvitz and his colleagues were interested in fates of cells. How do cells decide what to become during development? And the way they did it was by looking at the egg laying, because they could watch that. And when this structure, which is called a vulva, in the worm, it doesn't function right, the eggs don't get out. And they were able, therefore, just to line up a bunch of mutations that again fell into a pathway that gave rise in humans to cancer. This is the RAS-RAF pathway. It's the most common pathway activated in cancer. What you're seeing on the left is a patient with metastatic melanoma with the metastases spread throughout the body on a scan. And using this pathway that was worked out in the worm, just to understand cell fate during development. Using this pathway, a key node was found where a drug could interfere and reduce the cancer burden of this patient. And because we know that there are ways that cancers can get around a single hit, the next step was to find a drug that would hit the second part of the pathway that would make it impossible for the cancer to escape. And you can see this is a life survival curve. Patients survive much longer when you hit both of these. So we have a field now that started in the 70s based on fruit fly and nematode work, which are aimed at understanding the beauty of development. It was really not in any way linked to cancer. No one at the beginning thought that. I know these people very well. At their Nobel Prize speeches, they pretended they knew it was coming along, but they didn't know ahead of time. Okay? So this is the way all cancer drugs are discovered now, is to find the pathway. And what's so wonderful is these pathways, there's only maybe a couple dozen of these pathways that are involved in cancer. So it's a matter of, it's a way of organizing the whole genome to understand cancer. We no longer think of cancer by organ system. We think of it by pathway. Because you can have the same pathway activated in many different tissues, giving you different types of cancer. So there's no such thing anymore, really, as breast cancer. You should be knowing breast cancer, which pathway is activated. Okay, but I told you I was going to cure cancer. Does this cure cancer? Not usually. Hitting these pathways usually gives you a respite, but it doesn't last forever. Today, though, we have hope of curing cancer that comes from an addition to this of a different direction. And this one comes from trying to understand viral infections. Again, nothing to do with cancer. Turns out, and I'm not going to belabor this, when a virus invades a cell, that cell gets killed. The so-called T cell is part of your immune system that recognizes some part of the virus that's expressed on the cell surface and kills. That's how acute viral infections are dealt with. Viral infections can become chronic, though. And it turns out that they're chronic not because there's anything different in how the virus gets into the cell, not because it makes anything different, it still puts something on the surface, but because the cell that's infected makes a repressor that tells the immune system to stop. It's its own immunosuppressant. Now, a few years ago, folks said, is this very fundamental discovery relevant to cancer? Why should it be relevant to cancer? It's because cancer is actually foreign and should be rejected. Cancer cells are abnormal enough to always make abnormal proteins. They should be rejected by the immune system, just like a virus, but they're not. And so they began to wonder, what's the problem? Could it actually be this repressive shield that's put around the cells? And the answer is yes. And they found that antibodies that would block this repressive shield, for example, to PD-1 or CTLA-4, cause the cancers to go away completely and stay away, with some of these now over decades. So this usually will be added to the pathway therapies to give you a cure for cancer. And I do think that a cure for most cancers will happen in the next decade. It's a real cure, not just putting it on hold. So that's the first one. Second, I want to tell you briefly about editing a genome. This, again, came from extremely basic work. So you may not realize this, but bacteria get viral infections too, just like you do. But it's been known for many years that a bacteria, once it's infected with a virus, can't get reinfected. The question is, how does it know that? It doesn't have an immune system. It's one cell. So very fundamental work looked at it, and they realized that when the first virus gets in, it infects the genes in the cell, and then the bacteria makes a copy of that foreign virus and uses that to get rid of the virus. Not clear how that was going on. But the basic work that led to the potential therapeutic was the very fine structural analysis of how it did it. So what you'll see here is viral DNA. Let's see if I can show you here. That's coming in after a cell's been infected. The previous infection gave rise to this so-called guide RNA, and together they bring in this other protein, it's called Cas9, which cuts and destroys the virus. So you end up with a cut virus and very specific. So folks said, that's a really complicated system, but each of those pieces can be made in vitro. We could make those. Could we use those in humans? Could we use it in human cells? And the answer is yes. If you target any part of the human genome, you can actually cut it precisely at the place you want to cut it. And if you add a corrected gene, let's say there's a mistake, you can actually correct the gene function. So let's say you have sickle cell anemia, as 240,000 are born each year in sub-Saharan Africa with, or you have cystic fibrosis, or you have muscular dystrophy. In principle, one can go in now and repair that defect very precisely, either by cutting it out or putting in a new gene. So again, this work arose from a very fundamental problem of how does a bacteria get rid of a virus? And today there are at least 5 companies trying to get this into humans to correct any genetic defect. We're not there. We can do it in vitro. We can take cells out if they have sickle cell and put them back in after they've been fixed. What we don't know is if we can fix it in vivo in the person, but I'm sure that will come. So that's the second example. And the third is coming back, Congressman, to your point, is how can we reverse aging?
Hurry up.
Yeah, you're not alone, by the way. This crowd's too young to really get it, I think. It's So here's the question for you. Again, it's a very fundamental question. Is aging reversible? We don't really think of it that way. In fact, it's usually pictured as a ball like rolling downhill, and once you're an egg, and then you divide, and you go through gastrulation, and once you started to make your other tissues, it's all reversible. It's all irreversible is the way you think about it. It just keeps going down all the way towards differentiated state, and then ultimately those cells are programmed to die, and you die. So John Gurdon, back in the 70s, asked a very simple question: is it really true? Or could you actually reverse it? And he did a really, I think, a very clever experiment. He took a frog egg, and from that egg, which was fertilized, he got rid of the nucleus. So now this egg is just cytoplasm. It can't go anywhere. It can't develop at all. But he took a nucleus from the skin of another frog and put it. So now you've taken a differentiated cell, you're taking out the nucleus, should be only able to make skin cells. He did the same thing with gut, should only be able to make gut cells, and put it into this depleted egg and asked what happened. And he was clever because he took it from an albino frog. So he could be able to tell the difference of what was going on from the host frog. And it turned out that you reversed differentiation and you were able to get a whole frog. So you've taken a skin cell, then just the nucleus, all the genetics, you've reversed it, erased it, and gone down another direction. And he's able to do this many, many times. So it is reversible. And that's good news, of course. Now, it's even easier to do in individual cells in culture. That was done recently by just taking skin cells out and adding a few genes. We don't have to do the injection and the fancy stuff anymore. You can add 4 genes, and that cell goes back basically to ground state, if you choose the right genes. So it's basically back to a fertilized egg. And then by adding any factor, group of factors that have been worked out over years, we can make this cell become muscle, nerve, or bone. Now look at this. If I want to understand brain diseases, right? I'd like to understand the genetics of brain diseases. But there are very few people who are willing to give up a chunk of their brain, just as a matter of principle. Some people maybe wouldn't miss it, but whatever. But they think they would. But if you do this, you can do exactly the same thing with a human cell. So you take a human cell from the skin or any place, you can make it into a mini brain in culture. And in culture, what's amazing is not only will it retain all the structure of a brain and grow into looking like a brain, but it will retain the properties of the host it came from. So if you have someone who has a disease that we know by various studies will affect, for example, connections between cells or calcium moving in and out of a cell, that is still retained in the nerve cell in culture, even though everything's been erased and restarted and you've made a fresh mini brain out of it from zero. So, for example, I'll give you an example of something that I think gives us hope we can understand diseases now like schizophrenia or autism. On the left-hand side is what is shown. It's called a Manhattan plot. It shows all the genes that have been associated genetically with schizophrenia. And it turns out that they're all, in this case, related to the connections between cells. So this is one cell, another cell, and it signals by letting calcium in to the other cell. And you can see on the left here the signaling we can watch in culture of these cells. And the point of all of this is that the patient who had schizophrenia, the amount of calcium that goes in is actually less than normal. Now, why is this important in pharmaceutical development? This gives us an assay. We can say, can I actually restore this cell to normal, and then I can go into people. So this is extremely important technology for that. The last part I want to tell you about is comes back to this fundamental question, can we actually reverse it into human today? And it's important. In the early 1900s, about 4% of the population was over 65. Today, most countries, it's over 20%. And with that comes the onset of many diseases of aging. The worst in some ways is fragility. I mean, imagine if we actually could deal with weakness and hearing loss and loss of vision. You could even get people out of nursing homes, or you could get them out of hospitals. This is really a big problem. So what's the approach we're taking, and I'll finish with this, to this kind of fragility? Well, there's two different kinds. I call them, one's called an extended warranty and the other's spare parts. The extended warranty would be you just take a pill and you live a lot longer. And that was originally said for red wine, and unfortunately that's not true. But I still think the experiment hasn't been done enough. And there have been a couple of others. But it's unlikely we're going to find something and think about doing the clinical trial for 100 years. That's tough. But the spare parts piece is directed at looking at, can we repair organs by organ the aging tissue? And the reason we have hope comes from this very cute animal, the axolotl. The axolotl is amphibian that if you cut off a limb, it regenerates completely. You cut off a tail, it regenerates completely, and normally. That's not true for humans. So something is retained here that we've lost. And what it turns out is there's actually two things going on here. One is you have some stem cells that can become limb tissue for bone and skin and all of that in there. And the other is an ability to de-differentiate and go back and then redifferentiate down the limb. So we have now reason to believe from this very fundamental science that we could capture developmental programs even in the aged. And we've begun to focus on that. So take a program that usually plays out in the embryo and reestablish it. I'm going to show you one example that's for muscle weakness. About 10% of the population over 65 has such severe weakness it gets in their way of life. 50% have a hard time going up a flight of stairs over 65. So the way we do our work in general was to try to pick the most ferocious form of a disease and see if we could fix that, and then see if we could generalize it to the population. So I'm going to show you a particularly ferocious form of muscle weakness in a man named Joe, which we understood well enough to begin to make medicines to help him get better. And I'll let Joe speak for himself. I don't know how many noticed he's also a Patriots fan. So he's a good man. The pathway that controls muscle growth during development has been worked out on sort of a simplified version as shown on the left. Basically, it's a pathway that represses growth and has to be relieved. And then later in life, this becomes active and represses growth more permanently. So what we want to do is get a drug that will inhibit this pathway and allow muscle to grow. And when we did that, I don't know if you saw this. This is a football field. Folks like Joe were able to go and you measure how long they can go in 6 minutes and able to go more than half a football field farther. So this is, for them, a big deal. And we're working now on extending it in other areas, replacing cartilage in the knee, improving tendon strength, vision from macular degeneration. All of these are approachable by thinking about these as developing tissues. And all of that work came from very fundamental science. So my lesson is very simple. All drug discovery depends on basic science. It's a long and winding road. And what starts out just in an attempt to understand what's beautiful often end up with something that's very practical. Thanks very much.